The overall goal of the following experiment is to derive neuronal progenies directly and exclusively from pluripotent human embryonic stem cells using small molecule induction. This is achieved by adding retinoic acid to undifferentiated cells maintained under defined conditions. Next, the differentiation process is continued by detaching cells in a suspension culture where they form floating cellular clusters called neuroblasts. If neuroblasts are permitted to attach to a tissue culture plate in neural differentiation media, they develop characteristics of mature neurons. Ultimately, immunofluorescence and deconvolution or confocal microscopy can be used to show the effect of retinoic acid treatment in morphology and marker expression of treated human embryonic stem cells.
In other techniques, only a small fraction of cells pursue a neuronal phenotype, whereas our method allows well-controlled efficient induction of pluripotent human embryonic stem cells exclusively towards a neuronal lineage by simple provision of small molecules.
There are a few steps to be careful with when preparing stock solutions. Take care to slow thaw matrigel and human laminin from -80 degrees Celsius to 4 degrees Celsius overnight. Store media at 4 degrees Celsius and always warm up the media to 37 degrees Celsius before use. Also, when preparing human ES cell media, some of the supplements are added just before it is used. These include BFGF, ascorbic acid, human insulin, activin A,